So remember that nucleic acids are nothing but polynucleotides that are produced by using nucleoside triphosphates in polymerization reactions. So if DNA polymerases use deoxyribonucleotides in this process, we produce DNA or deoxyribonucleic acids. And if RNA polymerases use ribonucleotides, then we produce ribonucleic acids, also known as RNA. So we took a look at this picture last time, which shows that this polymerization process then produces strands that have directionality. Told you what this 5' to 3' directionality means. We have a sugar phosphate backbone, which is hydrophilic, likes to be exposed to an aqueous environment. That's important later also to understand the physical-chemical properties of nucleic acid. And then we have attached via the anomeric carbons of the sugars our nitrogenous bases, which are rather hydrophobic and don't like to be exposed to water. So I also told you that the nucleotide sequence or the base sequence actually then stores the information not only for the structure that may be assumed by such polymers, but also the kind of information that may then be translated into functional biological products. And I told you that for simplicity here we assume that this is a protein. Okay, so base triplet then would encode the information for particular amino acids that then can be utilized by ribosomes in protein biosynthesis. So let's take a look at the structures of the most common pyrimidine and purine nitrogenous bases that we find in RNA and DNA. So we'll see later that there are many variations of these structures. Actually, there are unusual nucleotides that we also find in particular in RNA, but these are the purine, the 2-purine and 3-pyrimidine bases that we usually find, especially as students. If we open a textbook, okay, we usually see adenine, guanine, cytosine, thymine, and uracil. I pointed out you should take statements like thymine can only be found in DNA with a grain of salt. This is not really true, as we will see. What you have to realize is that these nitrogenous bases are hydrophobic aromatic compounds, and with this come certain physical-chemical properties that they have. So what is an aromatic compound? What characteristics do aromatic compounds have?
Like a delocalized pi system?
Yes, usually a highly delocalized pi electron system. What does this mean, really, with regard to properties?
Make it acidic?
No, not really. We call them bases, right? Because we have these nitrogens that are electron donors. So they're Lewis bases, right? Where did we see a highly delocalized pi electron system before in a cyclic structure? Give me an example. Talk about this only recently. Heme, right? Look at the structure of heme with this protoporphyrin 9 system where we have highly delocalized pi electrons. So when you think of heme, what comes to mind? Look at your seat. Look at your seats. What's a property of these seats that you have? Not necessarily that they're cushioned. They're red, right? Okay, so when I think heme, I think blood, I think red. Okay, so why is heme red? Yeah, it's actually pi electron system, so that we can easily excite and then we produce color or see color, right? So it doesn't take too much energy to actually excite the electrons in this porphyrin ring system. So if we look at these compounds, we need a little higher energy actually to excite these electrons to a higher energy level. And nevertheless, we still have absorbance characteristics associated with these structures, and they absorb light, okay? But they do not absorb light in the visible range, okay? But they absorb UV light, okay? Because it takes more energy actually to excite them. But if we look at spectra of, in this case, nucleotides, we see that we have absorbance maxima for all of these around, let's say, 260 nanometers, to keep it simple. So all these nitrogenous bases and also the nucleosides and nucleotides have peaks, absorbance maxima in this range here. And this is quite similar to what we saw for the aromatic amino acids, if you remember that in my little review of amino acids, peptides, and proteins. I also showed you absorbance spectra of the aromatic amino acids, and we saw that especially tryptophan and tyrosine absorb around those wavelengths, okay, depending on whether you look at tyrosine and tryptophan, phenylalanine to a lesser degree. And I think I told you, or you should know by now anyway, that a particular wavelength is usually chosen to quantify or measure the absorbance of protein samples. Do you know what wavelengths that is? 280 nanometers, okay, and this is based on the absorbance maximum that we have for tryptophan. Tryptophan, we have actually some extinction coefficients of the molar absorption coefficients given here for our nucleotides, but the molar extinction coefficient for tryptophan is something in the range, if I remember correctly, of 5,500 or something like this. So if we look at the molar extinction coefficients here at 260 nanometers, is actually quite a bit higher compared to tryptophan, which is the strongest chromophore really in proteins. So this alone should make you understand that nucleotides have pretty strong absorbance in this wavelength range. And by convention, we use this wavelengths of 260 nanometers, not 280, but 260 nanometers, to actually quantify DNA and RNA samples, nucleic acids, and also nucleotides and things like this. Okay? So that's something that you may want to keep in mind. 280 is a very important number, because that's the wavelengths that we utilize for protein samples, and 260 is what we use for nucleotides and nucleic acids, and then you may also form the ratio, for example, to determine purity of samples that you have. There are programs that allow you to do that also. Okay, so another thing that you need to understand is that these nitrogenous bases are flat structures, okay, and I told you that they are hydrophobic. They can also interact through hydrophobic interactions, which we can call stacking interactions, and the stacking usually is compared to the stacking of coins. Okay, so if we have nucleic acids and we have bases next to each other, they can interact through these stacking interactions. There are also some dipole-dipole interactions, but usually we just refer to these interactions as stacking interactions. And when I look up pictures, Googled pictures of stacked coins, I came across some interesting structures that I usually like to share with my students, just to give them some idea that not everyone is spending their time studying biochemistry all day long, but looks like some people actually find time to build structures out of coins. And this is not particularly elaborate. If you look at these things here, this I believe is a radiator here on the wall, AC, so this is probably a structure that is at least that tall, okay. Think about it, okay, put these things together, one coin at a time, okay. So, these stacking interactions are not the only interactions that we can have between nitrogenous bases, and I guess you're all somewhat familiar with hydrogen bonding between complementary bases, and we'll talk about this in various contexts today and in the next couple of hours. But studies by James Watson and Francis Crick, actually in the early 1950s, elucidated that adenine likes to hydrogen bond with thymine. If we're talking about DNA samples, if we were talking about RNA samples, the thymine would be replaced by uracil, but we would have pretty much the same type of interactions here with adenine. Guanine likes to hydrogen bond with cytosine, as we can see here from these pictures. Adenine and thymine / uracil actually can form hydrogen bonds in these positions, 2 hydrogen bonds in these positions, while guanine can interact with cytosine via 3 hydrogen bonds, as shown here. So this preference of adenine for thymine, guanine for cytosine is actually based on steric and electronic complementarity that these molecules have. And this, as a result, then yields to double-stranded structures if we have 2 complementary strands, meaning that if in one position in one strand we have a cytosine here, we would find guanine in the complementary strand, in the opposite strand, the other strand, let's call it for the time being. If we have an adenine in one strand, we would have a thymine in the other strand. So we can form double-stranded structures. So such double-stranded structures can be formed either by 2 individual strands, or we could also form double-stranded structures if we have some self-complementarity within one and the same strand, and we'll see a couple of examples later. But Watson and Crick actually realized that a likely structure for DNA would be based on a double-stranded molecule. So nowadays we just take this for granted. Okay, but this was one of the biggest mysteries in structural biology and biochemistry the last century, and many famous people actually tried to come up with reasonable structures for DNA and did not succeed. Among them, for example, Linus Pauling. So Linus Pauling is one of the most important biochemists of all time, and he couldn't figure out the structure of DNA. He was around that time, actually 1954, he was awarded the Nobel Prize for elucidating the structure of another important molecule, namely peptides, and talking about the alpha helix, one of the most important secondary structures, and he could elucidate that structure, but he was unable to actually come up with a reasonable structure for DNA. But Watson and Crick gathered experimental results from people that had worked on DNA and put together models and did calculations, and then came up with a structure that made sense, namely a double-stranded molecule that in 3-dimensional structure and space actually assumes a helical structure, right-handed helical structure. We'll see a picture for that in a moment. But for example, some of these important experimental results that were obtained by other scientists that were taken by Watson and Crick were obtained by someone called Erwin Chargaff in the 1940s. And Chargaff actually had found that if one analyzes the composition of DNA taken from organisms, that there seems to be always the same amount of A, T, and G, and C. This on a basis. And this and other results were very important for Watson and Crick to actually come up with this double-stranded structure. And it makes complete sense if we look at the structure of DNA today, because for each C that we have in one strand, we would have a G in the other strand. For each A that we have in one strand, we would have a T in the other strand, and vice versa. Okay. But if you don't have no idea what the structure of DNA is, you can't really explain this result. So there are a couple of rules that Chargaff came up, but this equality of purine and pyrimidine base is probably the most important here if we look at the structure of DNA. So a couple of things about double-stranded molecules, and we'll focus on DNA for the time being before then later, or maybe next time we move on to RNA. So if we take a double-stranded molecule of DNA, the 2 strands are, as I said, complementary. Okay, so it's important to realize that they are not identical. If they were identical, we wouldn't have an A or a C here and a G here. Just assume a strand that has an A as the single nitrogenous base that it has in its nucleotides, then okay, we don't have all A's in the other strand, we have all T's, so they are complementary, they are not identical. Also, they are anti-parallel, meaning they run in opposite directions. So if we have the 5' end of the left strand here on top, and it runs down to the 3' end this direction, top to bottom, the other strand runs in the opposite direction. So they are complementary and they are anti-parallel. And in 3 dimensions, as I told you, they assume a helical structure, which here we see on the left in a ribbon diagram, and here on the right in a space filling diagram. So we are talking about a helix, almost like a helical staircase, that runs around an imaginary axis, okay? There's no axis that you would see here, but imaginary axis, and the direction is right-handed. Okay, so it's a right-handed helix. We'll see that also left-handed helices can be formed even in nature, but the classical so-called Watson-Crick double helix, which is also known as BDNA, is a right-handed helix. It has diameter, it's relatively narrow, of 2 nanometers, okay, but as I pointed out previously, it can be pretty long. Okay, and we'll see some numbers for this in a moment. So there are certain characteristics that are associated with molecules like this. You may remember this from the alpha helix or secondary structure in peptides. For example, the pitch, which is the rise per turn. So how much do you advance if you turn 360 degrees? So in this case, if we look at the BDNA, and you were very tiny and you would step up individual nitrogenous bases that you could use as steps, like in a staircase, okay, you would actually need to go up about 10 bases or base pairs, if we look at pairs of bases, and you would have advanced 3.4 nanometers. Okay. In structural biology, you will see not only usually nanometers as a unit, but also angstroms. Angstroms actually is just nanometers times 10. So structural biologists, that's my assumption, usually like to use somewhat larger numbers. So angstroms is just nanometers times 10. We'll also see both nanometers and angstroms on subsequent slides that I'm going to use. So the individual distance between adjacent bases is approximately a tenth of this, so 0.34 nanometers or 3.4 angstroms. Okay. With this knowledge, you could, for example, calculate how long a DNA molecule would be if it had hundreds of thousands or even millions of base pairs. You just need to know, okay, distance between individual adjacent base pairs is 0.34 nanometers. And for the next exam, I will also send out a study guide which refers you to questions in the study companion to the textbook, and there are questions like this, for example, how distance between, I think something like the distance between the Earth and the Moon is so many kilometers. How long or how many base pairs would a DNA molecule, fully stretched DNA molecule, have to actually reach from the Earth to the Moon, or something like this. Okay, just for you to understand. Okay, I can do this. I can calculate this by simply knowing this number. Okay, a couple of things. So there are minor and major groups that's based on the structural characteristics of our sugar phosphate backbone, and these groups are actually important for a variety of interactions with other biomolecules. For example, proteins like to bind in these groups and recognize certain sequences in DNA that we find. And if we have time, I doubt it, but if we have time, at the very end of the semester, I may talk a little bit more about the interaction of proteins with DNA, but probably due to the 2 snow days that we had, I won't have time to do that. In some years I have time to discuss this, in other years I don't. So I said that Watson and Crick actually elucidate the structure of DNA. So they published a landmark paper, a relatively short paper, actually, a little over a page only, in Nature in 1953, in April 1953. And I encourage you to take a look at this paper because it's so brilliant in its brevity, really. Such an important paper. And if you, okay, scientific paper really has to be lengthy and boring and complicated, but this really is right to the point and says everything that really has to be said. And they were awarded the Nobel Prize for this work. So I have highlighted a couple of things in this paper. So what they write is that the novel feature of the structure is the manner in which the two chains are held together by the pyrimidine base. Okay, nowadays we take this for granted. Okay, but they were awarded the Nobel Prize. And then they are joined together in pairs, a single base from one chain being hydrogen bonded to a single base from the other chain, so that the two lie side by side. Okay, good. And then they say also these pairs are adenine, which is a purine, with thymine, which is a pyrimidine, guanine, which is a purine, with cytosine, which is a pyrimidine. And then they also write that, however, if only specific pairs of bases can be formed, it follows that if the sequence of bases on one chain is given, then the sequence on the other chain is automatically determined. And again, you may smile upon this and think, oh, clear. Okay, you have to come up with this. Hindsight is always 20/20. Okay, but this has important ramifications, and this paper and this finding was so significant because it could easily explain other biological / biochemical phenomena that other models that had been postulated by the time had not achieved. Okay, and Watson and Crick summarized this really just in one rather matter-of-factly statement. Okay, this is very British. This is a typical British understatement, despite the fact that Watson is American. Okay, but he worked in England at the time. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. Okay, why? Because if you have one strand that has a certain nucleotide sequence, you can use this strand as a template to produce the complementary strand, to actually produce a copy of the other strand. Okay, and this is not only important for DNA replication when the genome of a cell has to be replicated before the cell divides into two daughter cells, but it's also important for transcription when the gene that is found on one strand actually is transcribed into an mRNA copy, an RNA copy that we call messenger RNA, can then be utilized by ribosomes for protein biosynthesis, utilizing the complementary strand as a template to produce a copy of the gene. Okay, and this is all just based on the simple fact that we have complementary bases that like to interact with each other. Okay, so here's a picture of Francis Crick, James Watson, and Maurice Wilkins. So Maurice Wilkins was an X-ray crystallographer, and you may know that he actually had a co-worker in his group, Rosalind Franklin, who actually studied the X-ray crystallographic structure, or she studied the structure of biomolecules, including nucleic acid, using X-ray crystallography. So this is probably a more proper statement. And Maurice Wilkins actually shared some of Rosalind Franklin's result with Watson and Crick without her knowledge, which is something that is, from a moral point of view, very questionable, should not be done. Okay, and unfortunately, Rosalind Franklin actually passed away before the Nobel Prize was awarded from cancer, and Maurice Wilkins actually shared the Nobel Prize with Watson and Crick. It doesn't diminish the accomplishment by these two gentlemen here, okay? But there has always been a question mark ever since, I guess, especially in modern times. okay, where ethics in science have become more, how should I say, have a greater awareness, let's say, in the scientific community. Okay, so the double helix is a rather stable molecule. Okay, so the two strands actually like to interact with each other, and we'll see in a moment also showing a schematic representation that if we have isolated single strands that are complementary to each other and we have them in solution under the right conditions, they really like to interact with each other again and form double-stranded structures. But this double strand is actually stabilized, here we see it, stabilized by a number of forces / factors or effects, the most important being the hydrophobic effect, which is based on the exclusion of water from the interior, so to minimize the interaction of our hydrophobic nitrogenous bases with the aqueous environment. So I can't remember if I mentioned the hydrophobic effect in this class. I probably did, but you should have learned about it in other classes like 470 or 472. So where do you find the hydrophobic effect, and where does the hydrophobic effect play an important role in the stabilization of certain biological structures?
Folding of proteins.
Folding of proteins, yeah, in particular if we think of globular proteins, spherical structure, spheroidal structure, and the hydrophobic amino acid residues are placed in the interior to minimize interaction with the aqueous environment, and we place more hydrophilic charged, at least polar side chains, preferably on the outside of the ball-like structure, so that these residues can actually interact with water molecules. Yeah, and this structure is then greatly stabilized really by this hydrophobic effect, which from a physical chemical point of view is a combination of different individual forces. But usually we just summarize this under the hydrophobic effect. So it's very important also for the folding of such proteins, okay, and then to stabilize these structures. And this by far is the most important effect stabilizing the double-stranded molecule. Okay. Nevertheless, we do have these stacking interactions between adjacent nitrogenous bases, which also further stabilize the structure. Then we have the hydrogen bonds. In some textbooks, you can read that the hydrogen bonds really are not of great importance for stabilizing the three-dimensional structure. Once we take a look at the next couple of slides, it becomes obvious that they certainly do have an effect. Okay, it may not be as important as the hydrophobic effect, but it certainly has a stabilizing effect of some importance. And then what we should not forget is that such a double-stranded molecule really requires the somewhat neutralization of the negative charges on our phosphates and the sugar phosphate backbone. And this is accomplished by having these negative charges interact with positive charges, which can either come from proteins, and we'll talk about the condensation of DNA molecules and the interaction with histones in a little while. But we can also neutralize these charges through interactions with, for example, polyamines. We talked about spermins, spermidines, and they interact with DNA molecules. They are positively charged through their protonated amines, okay? And then we also have interactions in particular with magnesium or manganese ions, cations. They can interact with our negatively charged sugar phosphate backbone. So this also plays an important role. So overall, as I said, double-stranded DNA is a rather stable structure, and we refer to this double-stranded state as the native state. And this is an analogy to the native conformation that proteins would assume under preferable conditions, to assume a certain three-dimensional structure that enables them to do their jobs, to be active. Okay, so here the native state would be a double-stranded state, but this native state can be destroyed, at least temporarily, by for example, heating up DNA samples, or also treating DNA samples with certain chemicals, and then we have this strand separation, and we transform our native state to the denatured state. So this is also called denaturation, an analogy to what we could do to proteins. Okay, if you denature a protein, for example, by heating it too much or treating it with some chaotropic salts, organic solvents or something like this, you destroy the preferred native structure, and you actually have a molecule that assumes, for example, a random structure that is not associated with the desired activity that this protein in the native state will have. So here it's similar. We can destroy the native state, for example, by heating up a sample of DNA. We actually provide enough energy so that these hydrogen bonds break, and then we have a single-stranded denatured state. But as you can see here, renaturation, so the assumption of the native state, actually happens relatively easily. Here it says special conditions required. This is vastly exaggerated. If you just heated the sample to separate double-stranded structures, then you just have to cool down your sample again. I wouldn't really call this special conditions. Now, if you actually use some chemicals to destroy the double-stranded state, then you might have to dialyze out these molecules, and then you would have a double-stranded molecule usually again. So special condition. I would erase this actually. It's not quite true. Okay. It sounds more dramatic than it is. But this shows us that, okay, once we have this complementarity in strands, okay, these strands like to form double-stranded structures. What you may know already is that the absorbance characteristics of individual single-stranded molecules are different from double-stranded molecules. Okay, and if we look at the spectrum in the range here between 180 to 300 nanometers of DNA sample, here in red the single-stranded denatured state, and here there's other, I can't even name this color, gray, blue, whatever it is. In the native state, we see that over the whole range here of wavelengths, the absorbance of the single-stranded molecules is higher, while overall the shape of these curves, the spectra of these molecules are pretty much the same. The absorptivity, so the extent by which these molecules absorb light in this wavelength range, is different. And we have a higher absorptivity here for the single-stranded state. So we have more absorbance, more color, and we can call this effect, for that reason, hyper.